In this activity, we're examining members of the Asteraceae family, such as sunflowers, daisies, and chrysanthemums. At first glance, these look like a single flower, but they are not. They are actually an inflorescence, meaning a cluster of many small flowers grouped together into a structure called a head. This arrangement is one of the most successful floral strategies in angiosperm evolution. Today, you'll separate what looks like one flower into its individual floral units. In composite flowers, what appears to be one large flower is actually many individual flowers packed tightly together. The outer petals are called ray flowers. The center consists of disc flowers. Each ray and disc structure is a separate flower with its own ovary. Below the head, you'll see green structures called bracts. These are modified leaves that support the inflorescence. Functionally, they are similar to sepals in protecting developing flowers. So when you count petals at first, you're likely counting entire flowers. This family is evolutionarily successful because grouping many small flowers into one head increases pollinator efficiency. Before observing closely, think about this question. Why would it be advantageous to group many small flowers together? Consider pollinator attraction. Consider energy efficiency. Consider seed production. Write your prediction before continuing. Begin by observing the entire head. Count what you think are sepals and petals. Now pause. If you counted more than 5 petals, you've discovered something important. Each petal is actually a ray flower. The central region consists of disc flowers. Locate the green bracts beneath the head. In your notebook, draw the entire head and label ray flowers, disc flowers, bracts. This drawing helps you visualize that this is not one flower, but many. Here is a ray flower removed. Observe its symmetry. Ray flowers are typically zygomorphic, bilaterally symmetrical. Notice that the petals are fused into a strap-shaped structure. Draw one ray flower and label the petals or corolla and bracts. Pause and complete your drawing. Now examine the disc flowers. Notice their developmental pattern. The most developed flowers are typically located toward the outer edge. The least developed are toward the center. Observe its symmetry. Disc flowers are typically actinomorphic, radially symmetrical. Notice the petals are fused into a corolla tube with 5 petals. In Asteraceae, stamens are often fused by their anthers. Locate the pistil. Determine whether it is simple or compound by looking at the stigma. Down below, look at the ovary. Is it inferior or superior? Where is the calyx? Is the ovary above or below it? The ovaries of the disc flower develop into the seeds you see in sunflowers. Now draw one disc flower in your notebook and label the major structures. Pause and complete your drawing. Now compare ray and disc flowers. How do their symmetry differ? How does their structure differ? What role do ray flowers play? Primarily attraction. What role do disc flowers play? Reproduction. This division of labor increases pollination efficiency. Write your responses using evidence from your observations. To summarize, a daisy, chrysanthemum, or sunflower is not a single flower. It is many flowers grouped together. The outer ray flowers attract pollinators. The central disc flowers perform reproduction. The inferior ovary is characteristic of this family. By grouping many small flowers into one head, the plant maximizes pollinator visits and seed production while conserving resources. This is evolutionary efficiency at its finest. And once again, structure supports function.